Chapter 23, we talked about how acetyl CoA, formed from each of our major biomolecules, is metabolized for energy in the citric acid cycle and the electron transport chain. In Chapter 24, we talked about how carbohydrates are taken into the body and can be used to supply energy through central metabolism or through fermentation in anaerobic conditions. We also talked about how they can be used to store energy. In Chapter 25, we're going to be talking about how triacylglycerols get taken into the body, digested into acetyl CoA for energy. We'll also talk about how acetyl CoA can be used to make fatty acids, cholesterol, and a special type of molecule called ketone bodies. Because we've already learned about hormones, we'll also be paying attention to how the new routes we introduce are affected by the hormones glucagon, insulin, and epinephrine. Just like with carbohydrates, we want to start talking about lipids in the form that they enter our bodies as food. Most of the lipids in food are triacylglycerols, so we'll focus on those for simplicity. We'll talk about how they get absorbed into our bloodstream as fatty acids, and the remainder and majority of this chapter will be more closely related to fatty acids and other metabolites. All of this is set against the backdrop of the citric acid cycle and the electron transport chain, which are the only ways that we get energy from fatty acids. There is no anaerobic metabolism for fatty acids. Just like with carbohydrates, we start triacylglycerol digestion by eating triacylglycerols. Unlike with carbs, we don't do anything with them in the mouth. This has everything to do with the physical properties of lipids compared to the physical properties of carbohydrates. Lipids are, by definition, insoluble in water. They tend to stay in large globs of fat that aren't accessible to enzymes until they get into the stomach. In the stomach, the churning action of the stomach breaks up those globs into smaller chunks called chyme. You can see these chunks if you vomit. I know at your home, but don't try this at home. The chyme has more surface area, and some enzymes, gastric lipids, can start acting on the lipids. Gastric here just means stomach. It isn't the substrate for the enzyme in this case. Lipases hydrolyze lipids, but only partially here. This is important because it means that at least some of the surface of chyme has become more polar. In the intestines, additional lipases can continue to work on hydrolyzing lipids, with aid of biolipids to improve their solubility. With many fatty acids free, their polar ends can interact with water and make anything unhydrolyzed more soluble by forming a micelle. Micelle structures, which you can see on the top left, are similar to intestinal cell membrane structures. There's a polar outside and a nonpolar inside. The polar outside of the micelle can interact well with the polar outside of a cell membrane and essentially adhere or stick right onto the membrane. The micelle can then open up the membrane and flip all of its fatty acids into the membrane, into the outer membrane surface. They can then be flipped to the inside of the cell by enzymes called flipases. They can be pulled into the cytosol and then packed up inside of those intestinal cells by reassembling into triacylglycerols and then packing them inside of lipoproteins and membrane lipids, essentially creating a big soluble cell-like structure containing the triacylglycerols. These are called chylomicrons. These chylomicrons are exported from the intestinal cells into the lymph system. They are too big to be absorbed directly into the bloodstream. The lymph system distributes these packets into the bloodstream and hydrolyzes them once they, again, into the fatty acids and some mixture of mono and diglyceride micelles that can then move freely through the bloodstream and be absorbed by cells. From there, they can be used or stored. This summary of lipid digestion makes clear the many transitions that triacylglycerols need to go through to make it into the bloodstream. This is different than other biomolecules because triacylglycerols are insoluble, and fatty acids only limitedly soluble. The triacylglycerols need to be physically broken into smaller bits, hydrolyzed to more polar fatty acids and mono and diglycerides, in order to have enough surface area to be absorbed into the intestinal cells. Then they need to be packaged as those chylomicrons to be exported into the lymph system and then hydrolyzed again to stay soluble outside of the chylomicron package. It's a lot of work, but the energy density of fatty acids makes it well worth our while, which we'll see later on in this chapter. Again, we can either store lipid energy as triacylglycerols, or we can use it directly. In most cases, since we're taking in lipids at the same time as we're taking in carbohydrates, that means insulin is going to be active. Insulin, as a hormone, is primarily responsible for promoting the uptake and use of glucose, meaning that it will trigger glycolysis as a major form of energy production, but slow down the use of fatty acids for energy. So most of our fatty acids are stored initially and then released later. Fatty acids are the major way that energy is stored in our bodies as triacylglycerols. Notice that glycogen can store some energy and blood glucose even less. That energy tends to be used up fairly quickly, and it tends to be distributed largely through muscle tissues, whereas triacylglycerols store a far larger amount of energy and localize it in cells called adipocytes that exist primarily to store fat. These cells will make up what we call adipose tissue, that are tissues that are rich in fat. These fats will be stored until we need them. When the glucose levels drops, insulin also decreases and glucagon increases, triggering the use of lipids for energy whenever possible. Similarly, epinephrine, as all the energy I can get right now, I need it, like, please give it to me type of hormone, will trigger the use of lipids for energy while continuing to use glucose as well. The release of triacylglycerols by any hormone is accomplished by a hormone-dependent lipase. As soon as the hormone is active, the lipase is triggered to be active, and these lipases will hydrolyze triacylglycerols back to fatty acids that can re-enter the bloodstream to be used by any tissue that needs them. This takes a lot longer though, so fatty acids are not really a good source for quick bursts of energy and are much more commonly used for long-term energy. Triacylglycerols contain 4 parts. 3 of those parts are fatty acids, and those are all tied together on one glycerol. The fatty acids are lipids and get used for energy in a lipid-specific pathway. The glycerol is actually really closely related to carbohydrates and can enter the 3-carbon phase of glycolysis to give us some small amount of energy. The fatty acid parts need a special pathway to produce energy, completely independent of glycolysis. This pathway is called fatty acid degradation, or oxidation of fatty acids, or catabolism of fatty acids. Sometimes it's also referred to as beta oxidation, which really refers just to the last part of this process, a cyclic set of steps that gives us all of the energy out of this process. The entire process of oxidation has 3 steps. The first step is an activation step. ATP is hydrolyzed all the way to AMP, that's adenosine triphosphate going to monophosphate, to give us the energy to attach a fatty acid to coenzyme A. Notice 2 things about this step. The first is that ATP is degraded all the way to AMP, which is equivalent of spending 2 ATPs, not just 1. The other is that the acyl CoA that is formed is essentially just a longer acetyl CoA. Acetyl CoA is just an acyl CoA with specifically 2 carbons. The second step is transport. Transport brings the acyl CoA into the mitochondria to be oxidized there. The acyl group is moved off of coenzyme A onto a molecule called carnitine that has a specific membrane transporter. Once inside, it moves back onto coenzyme A. And it's really important that the acyl CoA gets oxidized in the mitochondria because that means that all of the cofactors produced by that oxidation can be readily reduced in the electron transport chain for as much energy as possible. This is different than how glycolysis produces an NADH that then has to be transported into the mitochondria later, which costs us some of the energy we would have otherwise gotten. The third and final step of the oxidation pathway repeats a series of steps over and over and over again until the acyl CoA becomes shortened all the way to acetyl CoA. This recurring set of steps is called beta oxidation. These first 3 of these steps follow a pattern that we've seen also in the citric acid cycle. First, an oxidation of an alkane to an alkene using FAD and yielding FADH2, then hydration to an alcohol, and then oxidation of an alcohol to a ketone using NAD plus and producing NADH. This pathway also produces the same cofactors that we saw in that portion of the citric acid cycle. FADH2 for the oxidation to an alkene, and NADH for the oxidation of an alcohol to the ketone. The fourth step of beta oxidation splits the two ketone molecules into two acyl CoAs. One of the acyl CoAs is specifically acetyl CoA, 2 carbons, and the other is a carbon chain, an acyl group that is 2 carbons shorter than it was before. At the end of this step, we have an acyl CoA 2 shorter than we started, an NADH worth 2.5 ATP, an FADH2 worth 1.5 ATP, and an acetyl CoA that's worth 10 ATPs. So for every 2 carbons we shorten a fatty acid by, we gain a total of 2.5 plus 1.5 plus 10 ATP, or 14 ATP. This pathway is only oxidative. There's no anaerobic pathway for fatty acids. It's helpful to think about the 3 steps of fatty acid oxidation, the activation, the transport, and then beta oxidation, in terms of how we'd use that to calculate the yields for an entire fatty acid. No matter what, our fatty acid will always go through an activation that will always cost us 2 ATP. The energy gain will be mostly through beta oxidation, the 3rd step, so we need to figure out how many times that beta oxidation occurs. Here we show stearic acid, which has 18 carbons. The first beta oxidation gives us an FADH2, an NADH, an acetyl CoA, and a 16-carbon acyl CoA. So we've gone from 18 to 16. The next time we go through beta oxidation, we have FADH2 produced, NADH, acetyl CoA, and now a 14-carbon acyl CoA. So again, going down by 2 every time. This repeats 8 times. After the 8th beta oxidation, the 2 acyl CoAs that are produced are both acetyl CoA. When we go from 4 down to 2, it splits into 2 acetyl CoAs because a 2-carbon acyl CoA is acetyl CoA. So we don't need to go through beta oxidation for those very last 2 carbons. They just enter the citric acid cycle. So the overall yield of ATP for stearic acid is 8 times 1.5 ATP for the FADH2s produced in those 8 beta oxidations, plus 8 times 2.5 ATPs for the NADHs produced in 8 beta oxidations, and 8 times 10 ATP for the 10 acetyl CoAs produced in 8 beta oxidations. If you're looking at the calculation on the bottom, notice there's also a plus 10 ATP. That plus 10 is for the acetyl CoA that doesn't need to go through beta oxidation because it's already acetyl CoA. And then the minus 2 ATP at the very end is for that activation step. If you add all this together, we get 120 ATP for one 18-carbon stearic acid molecule. We can calculate this total energy yield for any saturated fatty acid as long as we can figure out how many beta oxidations occur. Since each beta oxidation consumes 2 carbons, we take the total number of carbons, in this case 18, and divide by 2. For stearic acid, this gives us 9. 18 divided by 2 is 9. 9 2-carbon pieces. However, we don't do beta oxidation on the last 2-carbon piece. It's always already acetyl CoA. So we have to subtract 1. So we take 18 divided by 2, that's 9 2-carbon pieces, but we subtract 1 from the number of beta oxidations we actually go through. So this gives us 8 beta oxidations. 8 beta oxidations, which are each worth 14 ATP apiece, plus 1 extra acetyl CoA worth 10 ATP, minus 2 ATP for the activation step, gives us a total of 120 ATP. So a different way of looking at that same calculation. This is worth comparing on a per carbon basis to glucose. Stearic acid gives us 40 ATP, 120 divided by 3, for every 6 carbons, while glucose only gives us 30 ATP. Longer molecules will give us even more energy per carbon. So fatty acids store energy much more efficiently than carbohydrates. We can do all the same calculations we just did for an unsaturated acid, except that every time there's unsaturation, we need to subtract an FADH2. This is because if we already have a double bond, we can't get the FADH2 associated from its production. We often need to move the double bond, which we can see in the second and third images on this slide, but that doesn't really change the energy yield at all. So for example, if we had 18:2, a fatty acid with 18 carbons and 2 double bonds, we'd get the same energy we would for 18:0, which we just calculated as 120 ATP, minus the energy from 2 FADH2s. In other words, we'd get 120 ATP, that's from the 18 carbons, minus 2 times 1.5 ATP, that's from the 2 FADH2s that we do not actually produce because of the unsaturation. This calculates to 117 ATP from 18:2. So we know that triacylglycerols can store energy much more densely than glucose, even when they're unsaturated, which makes me, and maybe a few of you, wonder why we have glucose at all. Fatty acids clearly store energy more densely per carbon, and even more densely when you take into consideration that the triacylglycerols don't need to be surrounded with water in the way that carbohydrates do. However, fatty acids are really difficult to mobilize. They're stored in specialized tissues, separate from where often our energy need is, and they need to be activated to release the fatty acids. And then they need to get to the right tissues. They make really good energy supply for functions that are always on, or for long-term use, but they make terrible surge energy. So skeletal muscles might use fatty acids to supply resting energy, they do, but they will switch to glycogen anytime they are being active. Similar, cardiac, that's heart muscles, can use fats most of the time, since your heart should always be on. But when it starts to work harder, it can use other energy sources as well. The liver, on the other hand, always prefers fatty acids and can use fat-based energy to help support glucose-based functions elsewhere, most notably in the Cori cycle that we saw in the last chapter. The brain poses the most interesting application of fatty acids. Your brain should be always on, but your fatty acids can't cross the blood-brain barrier, which prevents their use to supply brain energy. Your brain prefers glucose, but can also use a special metabolite of fats called ketone bodies to deliver energy from fats when glucose is limited. Since fats are unable to supply energy for the brain, but are likely to be the main energy source during periods of fasting, it's important to have some way of getting fatty acid-based energy into a form that can pass through the blood-brain barrier. We can't make glucose from fatty acids because there's no route from acetyl CoA back up to pyruvate. Instead, we make a class of compounds called ketone bodies. There are 3 ketone bodies, all shown in the top of this slide: acetoacetate, beta-hydroxybutyrate, and acetone. They're all initially produced as acetoacetate, the leftmost molecule. Beta-hydroxybutyrate in the center is just a reduced form. Notice the change from a ketone to an alcohol. This is formed with the aid of NADH, which is shown on the bottom left. The rightmost molecule, acetone, is a decarboxylated version of acetoacetate. That means carbon dioxide has been removed from it. Acetone isn't actually a really good source of energy. You can use it a little bit in the liver, but instead helps to balance drops in pH that are associated with ketone body use. The process of making ketone bodies is called ketogenesis. Based on the name, is the process anabolic or catabolic? There are several steps to make acetoacetate, which are essentially just joining acetyl CoA pieces together. 2 acetyl CoAs are added together initially, which you can see in the first row. A third acetyl CoA needs to be added to actually separate out the acetoacetate, which you can see in the second row. However, it gets recovered, and which you can see in the third row. The acetoacetate then is worth 2 acetyl CoAs, or about 20 ATP. Acetoacetate can also be reduced to beta-hydroxybutyrate after it's formed, which is another ketone body. Beta-hydroxybutyrate can be oxidized back to acetoacetate and form NADH, so it's essentially storing up energy from the NADH in addition to the 2 acetyl CoAs. It's essentially worth 22.5 ATPs, 20 from the 2 acetyl CoAs and 2.5 from the NADH. This slide doesn't show us the actual molecules, but it helps to get a general way to think about how the ketone bodies are formed, essentially storing the energy from two acetyl CoAs, and in the case of beta-hydroxybutyrate, also storing the energy of an NADH. When we need to use acetoacetate, or beta-hydroxybutyrate, we need to get it back onto a coenzyme A molecule before we can split it up into the two acetyl CoAs. In order to do that, it swaps places with a succinyl CoA, which is already on coenzyme A. Succinyl CoA is produced in the citric acid cycle, but normally when it's produced there, that CoA is hydrolyzed in order to phosphorylate a GDP and produce a GTP. It can't do that when it's used to process the ketone bodies, so we lose the equivalent of about 1 ATP when we're metabolizing ketone bodies. It's a small price to pay to feed our brains, though. The real problem with ketone bodies isn't the really small loss of energy. That's a small problem. A much larger problem is that acetoacetate and beta-hydroxybutyrate are the conjugate bases of acids, and when they're produced, they are going to drop the blood pH because they are acids. When the ketone bodies are present at elevated levels in the bloodstream, it's called ketosis. A slight case of ketosis will drop the blood pH slightly. A severe case drops the blood pH severely. Recall that with changes in pH, enzymes are no longer likely to fold as well and will drop their activity rate as they move away from the optimum pH. So whenever we produce ketone bodies in large quantities, this has a cascade of other effects on the bodies. One of these effects is that hemoglobin doesn't carry oxygen as well as it once did. In mild situations, this can be solved by eliminating some excess acidity by converting acetoacetate to acetone and carbon dioxide, which you can see on the bottom left. Both of the acetone and the carbon dioxide can be excreted through our lungs. Acetone can also be processed for a small amount of energy in the liver as well. That whole process is normal and happens to some extent when we're not eating for a while, like when we're sleeping. It's not a big deal. You can actually smell the acetone on people's breath if they're going through diet changes sometimes. And I can smell it on my kids' breath every morning before they eat breakfast. It's endearing, except, when it means they're hangry too.